Today we'll talk about the auditory system, which is responsible for hearing. Like other special senses, auditory system also has two parts, peripheral part and central part. Okay? The peripheral organs for the auditory system are the ears. You have two ears. Those are the peripheral organs. Now, the ear, only a small part of your ear you can see from outside. Most part is what? Inside. You don't see. Okay? So, the whole ear is divided into three parts. External or outer ear, middle ear, and inner or internal ear. Only a part of external ear you can see from outside. Which part you can see from outside? You can see the pinna. This is a part of the external ear. This is the external ear. Okay? So, pinna has a helix. This is the helix, and then you have the auricle. Auricle is the pinna, whole thing. Helix is this, outer border, and lobule is this lower part. Those structures you can see from outside. Then the outer ear canal, or external acoustic meatus, is also a part of the external ear. So, the pinna captures the sound waves. Sound is actually waves, right? Many waves come one after another. So, sound waves are captured by the pinna and funneled into the external acoustic meatus or outer ear canal. Then you have the middle ear. Middle ear is located inside the temporal bone. You know the temporal bone here. So middle ear is a cavity inside the temporal bone. And in middle ear cavity, you have three ossicles, tiny bones. Okay? And two small muscles. Okay? So, in the middle ear cavity, which is located inside the temporal bone, it has three ossicles or tiny bones and two small muscles. And the eardrum, this is the eardrum, also called tympanic membrane. So eardrum is called tympanic membrane, which is actually a partition between external and middle ear. In some books, you will see they have included it in the middle ear, some books in external ear. I would prefer to say that it is a partition. It is a partition between external and middle ear. Okay, so this is the middle ear. You see three tiny bones, malleus, incus, steps. We will see that later. And two muscles are here to keep these ossicles in right place. Then this part is the inner ear. Inner ear is the most important part of the ear. Why? Because you see a snail-shaped structure, snail-like structure, coiled structure, that is called the cochlea. Cochlea contains the sound receptors. Remember, the structure that contains the receptors, that is the most important structure. For example, in your eye, photoreceptors are located in what? Retina. Remember, retina has the photoreceptor. That's why I mentioned that retina is the most important part of the eye, right? Similarly, in case of auditory system, sound receptors are located where? Inside the cochlea. Make sense? That's why this is a very important structure. If the cochlea is destroyed or damaged, what will happen? You won't hear anything, right? So you have to sometimes do cochlear implantation, probably you have heard, right? Artificial cochlea is implanted. So anyway, that coiled, hard, snail-like, bony structure is the cochlea. Cochlea contains what? The sound receptors called the hair cells. Sound receptor cells or hair cells located where? Inside the cochlea, okay, in the inner ear. Also, you have two other structures in the inner ear. This part is called the vestibule, and these are semicircular canals. You'll see in next following pictures too. So those are three structures in the inner ear: cochlea, vestibule, and semicircular canals. Here you see this is the middle ear. Three ossicles. First one is attached to the eardrum, inner side of the eardrum or tympanic membrane. That first one is called the malleus. Then middle one ossicle is called the incus, and the last one is the steps. And those three ossicles are attached to each other like this. Okay, three ossicles are articulated or attached to each other. Now what happens, you see, sound is many waves, right? Create the sound. So sound waves enter into the ear through the external acoustic meatus and hit the eardrum or tympanic membrane, which is a very thin membrane. Okay, it's like, you play the drum, so it's like a membrane, and sound waves come one after another, right? And very high frequency. Sound waves are very high frequency. Like human sound wave frequency that we listen is 20 hertz to 20 kilohertz. Now, 20 kilohertz means what? Kilo means 1,000, right? So, in each second, 20,000 waves enter into the ear if you listen 20 kilohertz sound. Make sense? In each what? Second. 20,000 waves, right? If I say 20 kilohertz sound. If I say 10 kilohertz sound, you are listening, that means 10,000 waves are entering into the ear in 1 second. Make sense? Anyway, so sound waves are going one after another, right? In very high frequency and hitting the eardrum. So that vibrates the eardrum, right? So eardrum will vibrate in same frequency. Make sense? And now see, in the inner side of the eardrum, you have those three ossicles, right? And they are connected to each other. So what will happen? They will shake like this, right? Those three ossicles will shake like this, okay? That amplifies the sound pressure. So write down that three ossicles are helping to amplify the sound pressure. That is very important. Many times. They work like lever. You know, when you will see the big construction going on, have you seen the lever? So these three ossicles work like that. So it can amplify the power, the pressure of the sound. Okay. Then you see the last ossicle steps is attached to a window, and that window is called the oval window, oval shape. You see here, this area is oval shape. So a membrane is there. So steps is attached to that membrane, oval shaped membrane. That's why that is called the oval window. So oval window also shakes or moves in high frequency. So this is the last ossicle steps, and this is the oval window. It is a membrane. Okay. So oval window, and this is the steps. Okay. Now, from the oval window, the cochlea starts. The shape of cochlea is a snail-shaped bony structure. So this is the cochlea. Okay? And cochlea is filled with fluid. So now I can say that cochlea is a snail-shaped, coiled, bony structure filled with fluid. Okay? So, fluid. Okay. So what happens, you see, when the ossicles shake, the last one, steps, moves the oval window like this. So, in the other side of the oval window, you have the fluid. So, waves are created in the fluid. Makes sense? Waves are created in the fluid inside the cochlea. Now, if I make this cochlea straight, I can do it in the lab. I can take it, cut it out and make it straight in a petri dish. So let's do that. It's like a coiled, structure, so you can pull it and make it straight. So let's do that. So this is the cochlea. I have made it straight. So cochlea. Now if I see inside the cochlea, you have two membranes or partitions. Upper one is called the vestibular membrane. Okay, vestibular membrane. And the lower one is very important. That is called the basilar membrane. Okay. So inside the cochlea, you have two membranes. That means how many chambers? 1, 2, 3. Okay. 3 chambers separated by these two membranes, vestibular membrane and basilar membrane. On the basilar membrane, you have a very important structure resting on the basilar membrane, and that is called the organ of Corti. Remember, also called spiral organ. Same thing, okay? Some places you will see spiral organ, also called organ of Corti. Rests on the basilar membrane, and the hair cells are located inside it. Okay? So these are the sound receptor cells, hair cells. Why they are called hair cells? Because they have hair. Okay? Now, let's do something. Let's make a slice and take only this part out. Okay? So it has 3 chambers and 2 membranes inside the cochlea. So if I just make a slice from here out and see under the microscope, it will be like this. Right? 3 chambers, 1, 2, 3, 2 membranes. Vestibular, this one, and basilar, this one. So this is basilar membrane. Upper one is the vestibular membrane. And all these 3 chambers are filled with fluid. I mentioned it before. Everywhere you have fluid. And on the basilar membrane, you have the organ of Corti. Very important. So let's draw that one. So this is the organ of Corti. Organ of Corti. And I also mentioned inside the organ of Corti, you have the hair cells. So these are the hair cells. And these are hair. On the hair cells, you have the hair. So hair cells inside the organ of Corti. I can draw the nucleus inside if you want. These are the cells, sound receptor cells. Okay, very important. If these cells are destroyed, you won't listen. One more thing, on the hair cells or on the organ of Corti, you have a jelly-like soft membrane. And that membrane is called Tectorial Membrane. So you need to know this one too. On the hair cells, a soft jelly-like membrane is present, that is called Tectorial Membrane. Okay? So that is the structure of the cochlea. If I ask you, let's quickly review. What's the shape of the cochlea? Snail. Hard, coiled, bony structure filled with what? Fluid. And if you cut the cochlea, inside the cochlea, you will see how many membranes? 2. Upper is the vestibular, lower is the basilar, right? If there are 2 membranes, how many chambers? 3. Okay? All are filled with fluid. Okay? Now, on the basilar membrane, you have a very important structure. That is the organ of Corti. Why that is the very important? Inside you have the hair cells, sound receptor cells, right? And hair cells have what? Hair. On the top, right? That's why they are hair cells. Is it clear? Okay. Now, on the organ of Corti, you have a soft jelly-like membrane, like a structure that is called the tectorial membrane. Is it clear? Okay. So now look at me. This is a hair cell. Okay. These are hair. Okay. And you have the tectorial membrane on the hair cells, right? And tectorial membrane is soft or hard? Soft, right? So what happens? The hair of the hair cells are inserted into the tectorial membrane. Goes inside like this. Okay. So this is the tectorial membrane. These are the hair like this. Is it clear? Now you remember I said that oval window shakes, right? Steps move, oval window shakes, and waves are created in the fluid of the cochlea, right? So now you see, this is the tectorial membrane. Here are inserted into the tectorial membrane. Now when the fluid moves, the hair cells move like this. If you put something on the wave of water, it will move like this, right? So hair cells move like this. Every time the hair bends, signal goes to the brain. So this is interesting, right? Every time what happens? When the cell moves up, hair bends. Hair bend, right? So ion channels get opened. You know sodium influx, depolarization. Remember that? Same thing happens. So when the hair bend like this, ion channels open, sodium gets in, depolarization occurs, action potential. Signal goes to the brain. Is it clear? And when gets straight, stops. Again bends, signal goes. Okay? So every time signal goes to the brain. If you listen 10 kilohertz sound, 10,000 times the hair bend. How many times? 10,000 times in 1 second. Just think that. How fast? Yes.
So the depolarization of the hair cells is due to the bending of the hair?
Bending of the hair, at the root of the hair, you have the channels. So when bends, that opens the channels.
And then if you were to ask us the membranes inside the cochlea, we would only put the basilar and the vestibular. We would not put the tectorial, even though it's inside.
If I ask you, depends how I ask you. If I ask you which membrane is soft jelly-like membrane,
Okay.
you won't say that it's basilar membrane. You'll say
But if you
tectorial.
said, what are the two membranes?
If I
we would
say,
put the two
yeah,
main
if I
ones.
say which two membranes separate inside the cochlea into 3 chambers, you will mention those two, okay? Upper is vestibular, lower is the basilar, okay? If I ask you on which membrane the organ of Corti is located,
Mhm.
basilar membrane. So it depends how I ask the questions, right? If I ask you hair are inserted into which membrane?
Mhm.
Tectorial membrane, the soft one, okay? So you need to know everything, what I said, okay?
Mhm.
Good. So every time here bend, ion channels open, depolarization on. Okay? Depolarization means electrical signal, okay, going to the brain. Okay, so that is the mechanism. Now, who takes the signal from the hair cells towards the brain? These are the axons of the hair cells. So when hair cells produce action potential depolarization, signal gets out through the axons. And the axons form a nerve that is called the cochlear nerve. Makes sense? Comes out from the cochlea. So it is called what? Cochlear nerve. When you saw the ear model, did you see the ear model last week? Yes. You remember 2 nerves, one from cochlea, another from the vestibule, together called vestibulocochlear nerve, which is auditory nerve, cranial nerve number 8. Okay? So this is the cochlear branch of the auditory nerve, cochlear nerve. Okay, so that is what happens. Now, sometimes it can happen, these 3 ossicles are attached to each other. In these joints, calcification occurs, and that restricts the movement, flexibility of those joints. Make sense? Calcify. So sound will not be amplified. Make sense? Because I told you by the movement of those 3 ossicles, what happens? The sound pressure is amplified, right? So if that sound pressure is not amplified, since those ossicles are not moving anymore, so the pressure, sound pressure will be very low, and the person will lose the ability to hear in great extent. Make sense? If pressure is high, you will listen better. If sound pressure is low, your ability to listen will go down, right? So that is called athero. It will come. Okay. Another clinical condition that happens in middle ear cavity, if infection occurs in the middle ear cavity, that is called otitis media. Itis, itis is inflammation. So, otitis media, in the medulla, infection or inflammation. Okay? Okay, so if calcification occurs in these joints, that is called otosclerosis. Okay, otosclerosis is the calcification in the joints between the ossicles, and that causes hearing problem. Otosclerosis. Okay. Now, cochlea has the sound receptor cells or ear cells, so this is responsible for sound signal. Why you have the vestibule and semicircular canals? These two structures are responsible not for sound, but for what? Balance. Okay. Now you know, some of you, I'm sure, that if any problem occurs in the ear, inside the ear, sometimes the person gets vertigo. Have you heard that? Vertigo? So these structures are responsible for the balance of the body. How they work, just see. This is a semicircular canal. It is partially filled with fluid, not fully, partially. Now think that you take a tube and fill it with fluid partially. What will happen? If I move this way, fluid will go this way, right? If I move this way, fluid will go that way. Fluid will move. So those structures, semicircular canals, are partially filled with fluid, and they have sensors inside. Now if I put a sensor inside it, when I tilt this way, fluid will move this way, right? The sensor will be able to tell which way the fluid moved. Same thing, now look at my head. I have those canals inside the ear, right? So if I tilt my head, fluid will move, right? So the sensors will be able to tell which way the fluid moved and send signal to the brain that head has been moved this way. Make sense? If I move this way, the fluid will move this way, right? So the sensor will be able to tell the fluid moved that way and will send signal to the brain. So that's how your brain maintains the balance. Now if they are destroyed, then what will happen? The person will not be able to tell. Brain will not be able to tell which way the body is moving, head is moving. Is it clear? So that is the reason of vertigo. Loss of balance. I told you middle ear cavity is inside the temporal bone. So this is the temporal bone, and these are three ossicles: malleus, incus, stapes. And I also mentioned there are two tiny muscles. Remember that? What are those two muscles? One is called tensor tympani, and another one is stapedius. So you can see here stapedius, okay, and tensor tympani. And you see these muscles are attached to those ossicles to keep them, support them, okay? To do what? Support the ossicles. Now you see these muscles are tiny, but they play very important functions. If you suddenly hear a big, loud sound, okay, like an explosion, that sound has high pressure, right? We'll try to move the ossicles heavily because that pressure is very high. So ossicles will move very, a lot. But these muscles are supporting them. So we'll not let the ossicles move too much. Make sense? Limit the movement. In which condition? When you listen a loud, very loud, high, pressure sound. That, protects your ear, because those tiny muscles will not let the ossicles to move too much. Okay? Then they will get detached from each other, so the person will not be able to hear. Okay, so basically I have explained all of these things. Fluid. Now, what are those three chambers? Scala vestibuli, above the vestibular. This is not difficult. You see, this is the vestibular membrane, right? So above the vestibular membrane, this chamber is called scala vestibuli. Scala media is the middle one, okay, and scala tympani is the lower one. So scala vestibule, scala media, and scala tympani. Those are three chambers. Okay. You see here now, the same picture I did draw here, this one. Three chambers, scala vestibuli, scala media, scala tympani. This is the basilar membrane, this is the vestibular membrane. On the basilar membrane, you have the organ of Corti, right? Inside the organ of Corti, you have the hair cells. And on the hair cells, you have that soft jelly-like membrane that is called the tectorial membrane. Remember? And the hair of the hair cells are inserted into the tectorial membrane, right? So when the sound waves are created, what happens is it pushes the basilar membrane up and down. So the hair will bend and get straight, bend, straight, bend, straight, okay? The signal will go to the brain. Okay. Here you can see in larger picture, these are the hair cells. Here, tectorial membrane. Okay. You can see the hair cells under the microscope from top. If you see from above, there are two types of hair cells: outer hair cells and inner hair cells. And outer hair cells form V or U-shaped arrangement. And inner hair cells are linear, like straight line. The last thing, what happens once the signal gets out from the cochlea through the cochlear nerve? Okay. First, the electrical signal goes to the superior olivary nucleus, and then inferior colliculus. So first the signal will be taken to the superior olivary nucleus and then to the inferior colliculus. You can write down one thing. This is in the medulla and this is in the midbrain. Okay? Inferior colliculus is in the midbrain. And from there, almost all sensory signals are taken to the thalamus, because thalamus is called the major sensory relay station, right? So the signal from the inferior colliculus, the sound signal will be taken to the thalamus, and then from the thalamus to the primary auditory cortex. Now you tell me, how many of you remember the primary auditory cortex? Remember? How about this side? Where is the primary auditory cortex? Why you are doing like this? You did, yeah, exactly. The upper part of the temporal lobe, right? You saw it in the lab, in the brain model, and you saw it in the lecture. Where is the primary visual cortex? The back, yes. Right, so don't do like this. Those are not the questions I didn't ask from something new. We mentioned that again and again, right? In the lab as well as in the lecture. Primary auditory in the upper part of the temporal lobe near the lateral sulcus, and primary visual in the back, right? So from the thalamus, visual signal will go to the primary visual cortex, and from the thalamus, auditory signal will go to the primary auditory cortex, okay? Insula, it's in the bottom of the frontal lobe, under the frontal lobe, yes. Yeah, primary gustatory cortex, yes. Okay, so that is the pathway, okay? Signal will first go to the olivary nucleus, superior olivary nucleus, then to the inferior colliculus, and the midbrain, then to the thalamus, and then primary auditory cortex. Medulla has nothing to do with balance. Cerebellum is balance. Yeah. So here you can see the cochlear nerve. First we'll go to the cochlear nucleus, then to the olivary nucleus, superior olivary nucleus, then the inferior colliculus, thalamus, primary auditory cortex. Hearing loss. There are two types of hearing loss. One is called the conductive hearing loss, another is called the sensory neural. So conductive hearing loss, and another is called the sensory neural hearing loss. Okay, now what is the difference? Conductive hearing loss is easy to fix. If I put a piece of cotton in your ear, you will not listen, right? If I put ear plug, sometimes you put it, right? You won't listen because the sound waves are not being taken inside. It's not going inside. So that is conductive hearing loss. Make sense? If I ask you to put your fingers in your ear canal, right? That is what? Conductive hearing loss. Make sense? If any fluid accumulates inside the ear, sound waves will not be able to pass through. So that is conductive hearing loss. Is it difficult to fix or easy? Easy, right? If you remove that block. If any tumor is formed, right? That will be conductive hearing loss. And relatively, sometimes, the wax accumulates, right? And gets, hard. That can cause conductive hearing loss, right? Sensory neural hearing loss is difficult to fix or not fixable. What happens? The hair cells, you remember the hair cells inside the organ of Corti? Those hair cells are destroyed. So if hair cells are destroyed, that is the sensory neural hearing loss. Non-functional. I should not say only destroyed, could be non-functional. They are still there, but not functioning, right? Or less functioning. So that is sensory neural hearing loss. If someone is exposed to high frequency noise for a long time, like if someone works in a factory where that high frequency sound is produced all the time, they must use what? Ear muffs, right? That is mandatory. Otherwise, that person will gradually lose the hair cells, sensitivity of the hair cells. Hair cells will not function. Okay? So that is sensory neural hearing loss. If you take a medicine in very high dose, like anti-cancer drug or penicillin, antibiotic, that can also destroy the hair cells. So some chemicals can also destroy the hair cells. So that will be sensory neural hearing loss, right? And one thing, you remember when I talked about the nose and tongue, I said the basal cells are there, stem cells can quickly produce new cells, right? That's why if you drink something very hot, cells are dying, new cells are being produced, right? You get nose infection, cells are dying, new cells are being produced. But if the hair cells die, there is no stem cell there inside the organ of cochlea. Inside the cochlea, you don't have the basal cells. So that's why it is a problem, right? If the hair cells are destroyed, only thing you can do, you can implant the cochlea. But that won't be like exactly your original cochlea, right? It will probably help you in some extent, but not fully, right? To recover the function. So that is a problem, yes.
What about the supporting cells that we have in the ear?
There are no supporting cells. Those are the hair cells that are attached to each other. Okay. Oh, supporting cells are there. The supporting cells, let me see. Supporting cells could be there. Yeah, there are, sorry. These are the supporting cells. But only the stem cells are not there. Supporting cells just to support, they're not that important. Okay, so tinnitus. Tinnitus is also called ringing in the ear. Ringing in the ear. Many people suffer from this. There is no source of sound, but the person or patient hears the sound, although there is no sound. Nobody else is listening. That means what? The sound is being created inside the ear. Okay? Sound is created inside the ear, not outside of the body, inside the body. Now, remember one thing, this is very interesting about brain. Your brain is sitting in a dark cavity, right? Cranial cavity is very dark. It has no way to see what is going on outside, but it can tell you what is going on outside, right? It can help you to see, listen, all those things. How? The brain only counts. It's called digital processing. How many times it is receiving the signal? Okay? If your auditory cortex receives 10,000 times in 1 second, it will give you the perception of 10 kilohertz sound. Make sense? It doesn't care from where the signal is coming. If I put an electrode inside your ear, in the, to the hair cells or organ of Corti, and send electrical signal there to stimulate the hair cells will send signal to the brain auditory. You will listen to the sound. Make sense? So brain doesn't know from where it is coming, outside or somewhere else. Make sense? So that is very interesting. Now, without any sound waves, if your hair cells get excited inside the organ of Corti, signal will go to the brain. Intrinsic activation. You know epilepsy? Have you seen epilepsy patient? Right? So the brain cells are producing abnormal electrical signal. So that is good. So cell can start to produce action potential without any reason, intrinsically. That can happen. That's not normal. That is abnormal. Okay? Only the heart cells should produce by itself electrical. But if your hair cells do that, then your brain will count that number and will give you the sound. Make sense? So digital processing. This is very interesting function of the brain. Like if I, when you eat something, put like sweet on your tongue, you have the receptors, right? Remember the gustatory receptor? They will send signal to the brain. Like sweet, example, produces 10,000 electrical signals. Sour produces 20,000. Make sense? Bitter produces 5,000. So brain only counts that. Brain doesn't know what is on your tongue. It doesn't have any way to know what is on the tongue. It just counts how many times it is getting the signal, and by counting the number, it is giving you the perception. So that's very interesting function for digital signal process. Anyway, so let's stop here.